Kia ora. Nau mai haere mai ki tangata. I'm Nick Waipara. Welcome to Rangahau Rising and today's kōrero from an amazing speaker. I see that the AI companion is videoing us, so I will start today's hui with a karakia timatanga to bless our hui today, and then we'll get it rolling. Kia hora te marino, kia whakapapa pauna mo te moana, hei huarahi mā tātou I te rangi nei. Aroha atu, aroha mai, tātou I tātou e, te hei Māori ora. So, nau mai haere mai, welcome to BA, Bioprotection Aotearoa's Rangahau Rising. I'm really excited today to introduce our speaker. But before we do, we're just a bit of housekeeping. You can, if you keep the chat on, we'll take questions at the end, but you'll be able to see links to the previous Rangahau Rising seminars and some of the articles related to today's kōrero. So keep an eye on the chat. And also, I forgot to highlight a Tukutuku app. So if you want to get the BA Tukutuku app from the App Store, make sure you do download it. And the world is your oyster. Anyway, without further ado, I'd like to welcome Dr. Nicola Sullivan, who's an entomologist in the Ecological Pest Management Group of Aotearoa, otherwise known as the Bioeconomy Science Institute. In 2026, Nicola successfully defended her PhD on spider biodiversity and crop ecosystems. And this was supported by a food transition to 2050 scholarship through Lincoln University, Plant Food Research and Bioprotection Aotearoa. So Nicola's research has been of integral importance as part of the Bioprotection Aotearoa's mahi. It's particularly exciting because the mahi Nicola's doing has been showing the role of spiders, aka she-lobs, play in pest suppression for future food production systems. So it gives me, please give a warm virtual welcome to Nicola. Nau mai haere mai. Thank you.
Kia ora, Nick. Thanks for that introduction. Right, I'm going to share my screen and put this into presentation mode. There we go. We're all good.
Yep.
Awesome. So kia ora tātou, ko Nicola Sullivan tōku ingoa, and like Nick said, I'm going to be talking to you about my PhD research that was aligned with Bioprotection Aotearoa on the role that spiders play in pest suppression for food production systems. So a bit of introduction. Spiders are the dominant, most abundant and most diverse natural enemies in agroecosystems. So they're of economic value due to their pest reduction abilities, particularly when we're thinking about low residue practices. But there's been very few spider studies done in New Zealand crop ecosystems. And before my PhD, there'd been no published spider studies in New Zealand horticultural orchard systems. But internationally, studies have shown that spiders contribute significantly to pest control. They do this by providing direct consumption of pests, so they eat the pests. They also provide non-consumptive effects by overkilling, partial feeding, wounding. And if you think about spider webs, they'll kill small pests that are often ignored by the spider, but that still removes them from that system. There is some intraguild predation, which is when spiders will feed on other spiders or other generalist predators, but it's generally considered that the costs are outweighed by the benefits. And on top of that, intraguild predation can be reduced by providing complex and alternative habitats. So thinking about that, increasing plant diversity has been shown to increase spider biodiversity. And it's been found that the more complex the branch structure, the more spiders are present. So if we think about this photo down the bottom, you've got native forest fragment in amongst a kiwifruit orchard. And so that native forest fragment is going to have more plant diversity and more complex branch structure. So you would expect that there would be more spider diversity and abundance compared to the monoculture of the kiwifruit orchard. This research aimed to align with the goals of TAIO, or environmental well-being, being a holistic integrated approach to kaitiakitanga. And Article 2 of Te Tiriti o Waitangi acknowledges and agrees to tino rangatiratanga of the whenua to Māori, so Māori retain authority, which includes sovereignty over the land of Aotearoa. There's also a richness in Mātauranga Māori, which I was very keen to learn more about. So I did this thinking about the Kuruadao paper, identifying 4 key aspects to engaging with Indigenous people, including early engagement, speak the language, co-develop a framework, and establish agreements, including recognising and acknowledging the traditional ecological knowledge and sharing and communicating the knowledge gained. So for my PhD, I had 4 objectives. They were what spider species are present in Aotearoa New Zealand kiwifruit orchard and adjacent native bush systems. Does the orchard native bush interface act as a hard boundary, or does spider diversity flow between the 2 systems? What contribution are spiders providing to insect pest management? And how will vibration-based insect pest management affect spiders' ability to capture prey in Aotearoa New Zealand orchard systems? So we'll just focus on that first objective first, to look at what spider species are present. And this work actually started off as a master's thesis, and then I upgraded to a PhD. So when I started my master's thesis, originally all the work was supposed to take place in kiwifruit orchards in Kirikiri, and this was aligned with our plant and food research in our Bioeconomy Science Institute project, where there was lots of other mahi and research going on in that kiwifruit orchard. But this was started in 2021, and if you take your minds back to 2021, we had the COVID-19 lockdowns and travel restrictions. So I wasn't able to get up to Kirikiri initially, and I really needed to start getting some data. So because no one had looked at what spiders were in any orchard systems in New Zealand, I went and looked at what spiders could be found in Waipara vineyards, because I could get to that from Lincoln. I was then able to get up a bit further within the South Island still to Motueka, so I looked at what spiders were in apple orchards there. And then eventually I was able to get up to Kirikiri and look at the spiders in kiwifruit orchards up there. So obviously by doing that, I confounded the geographical location with the growing system type, which was kind of an inevitable just thing to keep in mind from the situation that I found myself in. So what I did in those systems was I had transects that went from a native edge into the crop. So I started at 0 meters, then a 5 meter, 10 meter, 20 meter, and 50 meter sampling point. And at each of those points, I had these different spider sampling methods. So I had pitfall traps, which are cups that you bury into the ground so that it's flush with the ground surface, and you put a metal tent over the top to stop debris and rain and stuff filling the cup, and you've got some liquid in the cup so that you catch the arthropods that are crawling around on the ground. I also had sweep netting, so you literally have a sweep net that you just sweep through the interrows and catch the spiders that you've got in the net. And then I had active sampling, timed active sampling, and this was divided into 4 categories. So I had daytime sampling and nighttime sampling, because a lot of spiders come out at night, and then had ground sampling, which was everything below the knee, and aerial sampling, which was everything from knee height to reach, to how far you could reach. And that was timed, so at that point it was timed for, yeah. Right, so results from here. If we look at this graph, on the y-axis you've got the sampling methods, and then on the x-axis you've got the count per sample per site. And we've got total spiders, the percentage that were adults, total adult spiders, and total immature spiders. In the black triangles, we've got the Waipara vineyards, the red stars are Motueka apples, and blue circles are the Kirikiri kiwifruit. So some key points here. More spiders were caught in kiwifruit compared with apples and vineyards. You can see that here with total spiders. Sweep netting had a low catch overall. I didn't catch a lot of spiders with the sweep net, but I did catch some species that weren't caught with the other methods, so it was still useful. The pitfall traps caught the highest number of adult spiders, particularly in the apples and vineyards, and more immature spiders were caught using the active sampling methods. So at this point, I upgraded to a PhD and became more involved with bioprotection Aotearoa, because one of the main sites for bioprotection Aotearoa mahi was in Te Kaha. So I again looked at that first objective about what spider species are present in the growing systems, and also the second objective about the orchard native bush interface. Does it act as a hard boundary, or does spider diversity flow between the two systems? So field sampling was conducted in Te Kaha, up there in Tairāwhiti, Eastern Bay of Plenty. I had field sites which were kiwifruit orchards. The kiwifruit orchards were paired either with the native forest, that they'd border the native forest, or border maize. And there's a lot of kiwifruit grown in Te Kaha, and the other thing that's grown a lot of is the maize crops. So maize presented a high disturbance system compared to the native forest being a low disturbance system. And so I sampled right through into the adjacent crop for these ones, both going, at 0, 5, 10, 20, and 50 meters into those land use types. The same sampling methods were used, so pitfall traps, sweep netting, and active sampling. And I had 4 field sampling expeditions in Te Kaha in summer and autumn 2023 and 2024. So all spiders were identified to family, and the adults were identified to species level morphologically. In spiders, you identify the spiders to species by their genitalia, and their genitalia is not developed until they're adults. So you can't accurately identify all the juveniles until they're adults. And all spiders were labelled to museum standards, and they're now being housed in various museums around the motu. So some key results from that work. If you look here, the graph on the left is being explained by the factors on the right. So on the right, we've got various spider families. So the Staphylidae up the top of the graph there, that's your big sheepweb spiders, which Nick refers to as shelob. You've got your Tomicidae spiders, they're crab spiders. These are your orb weavers, your ground spiders, your long-jawed orb weavers. Your lycosids are your wolf spiders. Linophiidae, your dwarf sheepweb spiders. Salticidae are your jumping spiders. Therideidae are your cobweb spiders. And Cyclotenidae, I don't actually know what the common name for them is. But yeah, so over here on the left graph, you can see we've got the maize in yellow, and you've got the native in red. Then you've got kiwifruit that were adjacent to maize in green, and kiwifruit that were adjacent to native in blue. And what you can see is that these maize in the yellow here are all grouped together. And so they're being explained by these spider families here. And the native up here in the red are all grouped together, being explained by these spider families here. And so they're quite separate, quite distinctive spider composition by the maize and the native. And then the kiwifruit are kind of a mix in the middle. So the land use did really dictate what spiders were harbored there. This is a very busy slide. It's a busy graph, but we've got the value on the y-axis, and on the x-axis here, you can see these are the transect sampling points. So K stands for kiwifruit. So you've got kiwifruit at 50 meters through to the edge here, and M and N stands for either maize or native, again at that 0 out to the 50. And maize is in yellow, native is in red, kiwifruit next to maize is in light green, and kiwifruit next to native is in dark green. So some key things to point out here. Total families and immature spiders, more were found at the edge, at these 0 points, and this decreased towards the center of the crop out to those 50-meter sampling points. And this EVAR here is a measure of species evenness. And so species evenness was lower at the edges and higher towards the center. So there were higher unevenness at the edges and higher species evenness in the middle of the crops. So what we kind of think is happening here is that the immatures are arriving at the boundary, because the way that a lot of spiders will move is by ballooning. And this is when they put up a bit of silk and they get carried on the wind in the right atmospheric conditions, and they'll blow on the wind. And that happens a lot. A lot of species will only balloon when they're immatures. So we suspect that there's immatures arriving at the boundary from the neighbouring source, but it could be more to do with the conditions within the orchard, which determines whether the immature grows through to an adult or not. And that has been found in some other studies in other countries. So also you can see down here, these graphs are from the most abundant spider families. So Araneae, your orb weavers, Linophidae, your dwarf sheepweb spiders, Tetranathidae, longjaw orb weavers, and Theridiidae, your cobweb spiders. And you can see, as was shown in the previous slide, the most abundant families show a clear preference in land use type. Throughout this, there was engagement with mana whenua. So Bioprotection Aotearoa worked closely with Te Kaha Group, which is an ahuwhenua trust of Te Whānau Apanui. It's a collective of kiwifruit growers. And I also connected with the chair of the runanga of Te Whānau Apanui and continued that connection. And he was a key person for introductions, knows everyone there, including the principal of the iwi kura, which happened to be his daughter. And so I was able to offer outreach and offer the return of the spiders that I had collected there in vials as a learning resource. And then there was an opportunity through the Bioeconomy Science Institute to apply for funding. So I got funding to lead a project doing that outreach, and was able to include lots of people in that outreach team, particularly from plant and food sites at Te Puke and Hawke's Bay that are closer there. So this is on the third outreach expedition. This was our outreach team. And you'll see it included Finn, who was a bioprotection at Aotearoa PhD student who also did her research in Te Kaha. So that was very cool. And during that third expedition, one part of what we did was return these spiders in the vials as a learning resource for the kura. And that went along with this spider flip chart, which I made with a lot of help from Julia Palmer, who is also a Bioprotection Aotearoa alumni and subsequently got a job at the Bioeconomy Science Institute. And so we put together this flip chart with a whole lot of slides. This is an example of one of them, but just on a bit of information about common spiders and the spiders that were found up there in the rohe. And it's in both English and Te Reo Māori, and that was Huata Arahanga from Bioprotection Aotearoa who did the Te Reo translations. And this resource, so that the kura were very appreciative of this resource, and it's also gone wider. It's gone up to the Bioeconomy Science Institute staff to use, and that's also gone to the New Zealand Association for Science Educators and a new bilingual kura that is about to start up in Horswell and Christchurch as well. So that's a really cool output that's come from my PhD and involves other Bioprotection Aotearoa alumni. So looking at the third objective, what contribution are spiders providing to insect pest management? We come into the lab now. And for this, I used 3 spider species from different feeding guilds that were commonly found in my field sampling. So I had an Araneae, Socca pustulosa. So that's your garden orb weaver, and you're bound to have this in your garden, that classic orb weaver spider web that is likely to be this dude. Salticidae, those are your jumping spiders. So Muratus griseus, that's your white banded househopper. And again, you're bound to have this in your house, hopping along your windowsill. Becky's probably terrified at the thought of that. But anyway, the Dacidae, Bedumna longingua, again, this one, if you have like a messy web around your car wing mirror, that will be those spiders. And so then we had 2 prey items, passion vine hopper adults and leaf roller moths, the adults. Those prey items were selected because they are key pests in kiwifruit orchards, but also for their communication type. I'll talk more about that later, but yeah, the passion vine hoppers use vibrational communication and the moths are not known to use vibrational communication. So I housed them in these 1 litre plastic arenas with a water reservoir down the bottom and a plastic stick for the spiders that build webs to build web from, and had this 3D printed vent and feeding tube to introduce the prey items, and this is relevant later. So I starved the spiders for 2 weeks to ensure equal starvation levels, and then put 8 prey items in each spider container at day 0, and that number was based on some pre-trials I'd done a year earlier, and then checked each day for 5 days to see how many prey items had been consumed. So key results from that, all 3 of the spider species studied consumed both of the prey species studied. The Bedumna longingua by far consumed more than the other 2, and actually so much so that even though I'd done pre-trials to gauge how many I should give them, several of the Bedumna ate all 8 of the insect prey items after the first 24 hours. So I had to do a follow-up experiment later on with 20 prey items after they'd been starved again to get to try and max them out. And yeah, so the maximum that they ate after 24 hours was 9. And the maximum after 5 days was 15, and so they had an average of 10.4 passion vine hopper or 7.7 cob, which is the moth. And you can also see that all 3 of the spiders ate more of the passion vine hopper than of the moth. The 4th objective, how will vibration-based insect pest management affect spiders' ability to capture prey in Aotearoa New Zealand orchard systems? So I'll just give a bit of background around biotremology or vibrational communication. It's an emerging field of biology. It's not bioacoustics, so it's not your airborne sound. In the most basic sense, biotremology is the study of vibratory communication behavior through use of substrate-borne boundary or surface mechanical waves. That's a lot of words. It's considered one of the most ancient forms of communication, and it's used by a whole lot of life forms and has many functions, both intraspecific and interspecific. So a few examples just to help the understanding. One very relevant one is this spider web up the top. So a prey item gets caught in the spider web and vibrates that substrate of the spider web. And that is what the spider picks up on to know, okay, I'm going to go for this prey item. Down the bottom, we've got this elephant and ant relationship going on here. So these ants build these nests in trees, which the elephants eat the trees. So it's been found that if the wind is blowing, the ants are fine. They don't mind that. They'll just continue on their day. But if they feel the vibrations through the substrate of the tree of the elephant walking, then that's an alarm signal to get out of the way so these elephants don't eat them. Then on the right here, we've got intraspecific communication. So these are vibratory signals of the green veggie bug, Nazara virgula. And you can see there's very different calls. So female calling song looks like that, male calling song like that, female courtship, male rival. And before I started my PhD, I did some work to show for the first time that the passion vine hopper, this guy in the middle here, communicates via vibrations, and identified this male calling signal up the top and the female calling signal down the bottom. And the idea behind this biotremology is that we could emit the vibration signal through this mini shaker that goes down the cordon wires of the orchard and through the leaves and branches that are touching that cordon wire. So that mini shaker, if you think kind of like a record needle, that's how it emits those vibrations. And the idea is that if you put the passion vine hopper mating signal through there, you've got this broadcast mating signal. So the passion vine hopper then can't actually find a mate to mate with because the signal is just everywhere. So it sort of confuses them. And so they don't mate, and that lowers the population. But my question was, does this vibrational communication, this broadcast, have any off-target effects on spiders? Because spiders are known to also use vibrational communication. So to test this, I had the bioassay arenas again, and that's where this nut and bolt attachment with a 3D printed spacer attached these bioassay arenas to this aluminium disc. And I had 6 bioassay arenas equidistant around the disc, and then this mini shaker in the middle, which was emitting the passion vine hopper male calling signal. And yeah, so I had the same 3 spider species and the same 2 prey items, and vibration treatment and no vibration treatment. So you can see here, this is where the communication mode of the prey item comes into play, because we've got passion vine hopper, which use vibration to communicate normally, and the moths, which are not known to use the vibration. So a single prey item was fed into this feeding tube, and then the time to prey capture was recorded for up to 30 minutes. If they didn't catch after 30 minutes, it was recorded as did not catch. But then after 24 hours, we came back and recorded which prey item had been consumed or not. And this was repeated once per week for 4 weeks. So the main results from this experiment, don't worry too much about the soccer pustulosa. That didn't really work so well because I could only find juveniles in the field, which really represented what I found in the field in my field studies anyway, many more juveniles compared to adults, but it meant that they weren't really big enough to catch the prey item. So that was a little bit of a flop. But anyway, these other 2 spider species showed some really interesting results. So you've got this jumping spider here, which is an active hunter, right? It doesn't catch prey using a web. It pounces on its prey, jumps around and pounces on its prey to catch them. But these Bedumnal and Jinqua, they use a web to catch their prey. And we've got the moths in green and the passion vine hopper in pink and red. So you can see the moths, whether there's vibration in the light green or no vibration in the dark green, there's no significant difference in either of those spider species. But the passion vine hopper, which does use vibrational communication, if you have vibration on, then the jumping spiders caught fewer after 24 hours than if there was no vibration. However, in the Bedumna, if there was vibration, they caught more passion vine hopper than if there was no vibration. And what we think is going on here, if you think about the passion vine hopper, which uses vibrational communication, it has been shown that they are more likely to move with the vibration present. So if you're an active hunter trying to capture a prey item that's moving around a whole lot, you're less likely to be able to capture that than if there's no vibration, it's not moving around so much. But think about a spider which uses a spider web to catch its prey. If that prey item is moving around a lot when the vibration's on, it's more likely to encounter the web when it's in a confined space and get caught by the spider. Whereas if there's no vibration, it's not moving around so much, it's less likely to encounter the web. So yeah, there's this interesting 3-way interaction going on between the spider species, the prey species, and the vibration treatment. And it means that, bioeconomy is likely to have a small effect on beneficial spiders, but it can be either positive or negative depending on the species, which is likely related to the hunting strategy. So just to kind of summarize what I've talked about, I've created the first inventory of spiders in Aotearoa New Zealand horticultural systems. And if you want to see that in more detail, you can look at my thesis or in my published papers. Different land use types harbored different spider families, and there was a limited flow of spider diversity between the systems. The 3 spider species studied consume both of the 2 target insect species studied, demonstrating that spiders do contribute to insect pest management in Aotearoa, New Zealand. And there was a 3-way interaction between spider species, prey species, and vibration treatment, which means that the applied bioeconomy is likely to have a small effect, but it can be either positive or negative. And I've got lots of people to thank, lots of people who came out hunting spiders with me, some very fun and interesting nighttime expeditions, particularly with those who do not enjoy spiders so much. So thank you to those people for putting their arachnophobia to the side. People helped me collect spiders for my bioassays, and there's various people there. And just to acknowledge my supervisors as well, Cor, Lloyd, Amanda, and Nick. Thank you all. And my funding from Food Transitions 2050, the Bioeconomy Science Institute, and Bioprotection Aotearoa. So yes, thanks everyone, and I'm happy to take questions.
A virtual homai te paki. You can see that the screen's really going off. Well done, kia ora. Firstly, I'd like to acknowledge your mahi by recounting a whakatauki or proverb around your beloved pūngāwerewere or spiders. Te ara o tuku pūngāwerewere. And that's around the paths of undertaking highly complex and intricate work or mahi, like the spider who toils to build a web, working methodically through complex situations, leading to a beautiful result. And I thought that whakatauki really represents your mahi and your PhD and where you've got to. So that is really quite a apt whakatauki, I thought. So kia ora. I will, put your questions in the chat. He pātai e te tahi if you have any comments or questions. Working with, I'll just have a comment, I guess, to whānawa apanui, the iwi and the growers. Were your mahi or your research working within actively operating kiwifruit orchards and maize fields? Was there sort of an appreciation of the beneficial insects in their orchards already, or was there an excitement about, okay, let's see what do you find and what could be applied actively in say kiwifruit production? Was that sort of, did you get any feedback?
Yeah, definitely. And I was really pleasantly surprised at how interested and appreciative of spiders people that I talked to were up there. They sort of generally knew that they were good. And somebody said to me, Rob Ruha's wife said that it was a saying in their house that he pūngāwerewere are the kaitiakitanga, they're the caretakers. And so people were really interested in how they are there. And there was an organic farmer up there who was really keen to know if they had more spiders in their orchard than in others. And I had some excited people, like the people coming to show me there's a bird dropping spider, which if you don't know that, it literally looks like a bird dropping, and they're very cool, and they have these cool egg sacs. And so people were excitedly showing me this and asking what it was about. So yeah, there was definitely that appreciation there.
And I have to tell everyone, Nicola was a star in that rohe. Like, she was famous. So, like, you really did have an impact. There were, like, the whole community knew about your mahi and wanted to come on night walks with you. So, like, yeah, and you and the team continuing to work with kura, that must be a really, it's nice when you feel like your work and who you are is reflected in the community. So I tell you what, she's a megastar in that rohe. So just so you know. Um, I'll go to the questions and please put your partile comments in. And you can also see, thank you, Sarah. She's put the links to Nicola's research papers and interviews on her multimedia platforms, where she's been very wonderfully showcasing her work. Kia ora, Vaughan. Thank you for a great presentation, Nicola. Perhaps not a question, but I think your results are pointing us towards agroecosystems needing to move towards greater biodiversity to gain the greatest benefits, network connection, diversity, and biocontrol gains. So I guess that's a reflection there.
Yeah, totally agree, Vaughan. And I mean, we can really see that spiders contribute to that pest management. So they're a big part of it, and there's ways we can encourage them. And when we think about, there seems to be an indication that the conditions within the orchard are having an effect on what spiders actually stay there. We should think about, what that means and how we can reduce pesticides, but also keep on top of that pest management, which means that biodiversity and creating resilience, right? So yeah, thanks, Vaughan.
Ngā mihi, Becky. Oh, well, firstly, are these flip charts available for Becky?
Yes,
Yeah, for
yes, they
others?
are, Becky,
Awesome.
and I can email you that. So the PDF is available that I can send, but I'll also get in touch to see if I can send a hard copy to you to pass on.
And yes, the laughing of her predominant use of grass versus spider pictures. Very visual. Question: what would you recommend to industry if they wanted to increase spiders' productivity in their orchards?
Yeah, well, I think we need to be careful about what insecticides are being used. There's a fair amount of literature out there on the effect of different agrochemicals on spiders, and not necessarily mortality, but the effects on their prey capturability and ability to build webs and things. So we need to think about that. Also, having shelters for them is important. The spiders really care about the structure. They don't necessarily care about flowers or anything like that in terms of a pollen resource, because they don't feed on the plants, but they need those structures to be able to build their webs and to hide away. So there's been some studies done on like even just corrugated cardboard or little refuges being put in orchards, say around trunks, to increase the numbers of spiders there so that they can feed on those pest insects.
I guess related to that, I guess the industry groups like, let's say, Zespri or say New Zealand Apple and Pears, is that something this sort of research can help, I guess, their wider engagement and implementation of that sort of benefit into orchards? I don't know if that is something that they promote. I know that Zespri are very strict on what people can do in their orchards under
Mm.
license, but I mean, I don't know. Does that something that you found any industry sort of wanting to develop this further?
Yeah, I've sort of had indications like from business managers that Zespri would be interested in this, and also that a lot of vegetable growers actually are really interested,
Oh,
like see spiders as being important in pest management, and there's a lot of interest there about how they can enhance that. And I think for apples and pears as well, I think it's very relevant.
yeah, I think some people will be quite surprised the amount of biodiversity and actual growing agroecosystems as well. Actually,
Mm.
there's a lot of people that think they might look monocultural, but they're actually not. So I think your sort of research does remind people it's not all just tucked away in a national park. But um,
Yeah.
Of course, spiders are quite, I guess, how would you say, divisive. There's as many people love
Yeah.
them as are sort of nervous from them.
Yeah.
Classic arachnophobia, but you've definitely got some stars there.
Yeah.
And
Yeah, that's
greedy
right.
stars. I like the one that eats the passion vine hopper.
Yeah, that's right. I mean, gosh, they ate so many. It's like couldn't keep up. And yeah, so I try to promote the spiders as being, cute and fun. And I mean, I love doing that outreach with the kids because so many of them, like when I did the outreach with the kura, we had sort of 3 rotations of classes of about 30, and so many of them were interested in the spiders. There would only be like 1 or 2 per group that were a bit scared, and they were just so into it. And then we went out and hunted for spiders, and everyone wanted to, find a spider. And I think there's a lot about that kind of just creating that curiosity and that understanding that generally, particularly in New Zealand, spiders can do far more good than they can harm.
Mm. Wow. And some humans harm them more than, especially
Oh,
if
by
they
far.
fall on their webs and
Yeah.
in an attempt to help you sample. Uh,
Get caught in your hair. Yeah.
uh, so what's the future for spidery? I know this is a very esoteric question and future probing, but um, we have people are absolutely compelled by your mahi because there's no one questioning your error bars or your graphs or anything. So, oh, there we go, Vaughan. Yeah, exactly. Where next for your research? The future.
Yeah, well, one thing I'd like to look at is molecular gut content analysis of spiders because, the work that I've done is looking at how many prey items can be consumed, if we don't give them a choice. Like I just, put the prey items in and they either ate it or they didn't. But what are these spiders actually choosing to eat in the orchard? So you can do that by looking at molecular gut content analysis. That doesn't tell you how much of that prey item will be eaten, but it does tell you whether they're choosing to eat it or not. And I think in general, just taking some of this stuff to the field as well, some of the stuff I did in the lab, like the vibrational communications, the findings there, do they hold true when you're out in the field? Are there ways that we can work around that? Like the passion vine hopper are only around for 2 or 3 months of the year. So, you wouldn't have to have that vibrational communication out for them all the time. Yeah, just taking it further and how can we actually operationalize these findings?
And also this whole innovation around biotremology, opening
Yeah.
that opening that innovative area. I mean that I guess all sorts could be done that way.
Yeah,
Like
that's
all
right.
sorts of future research, not just related to spiders, but that's quite a,
Definitely.
I mean, that must be something that's not that common, at least, or is it? I mean, it seems very, it's great that it's something that can be looked at in terms of like studying biodiversity and other things.
Yeah, that's right. It shows a lot of promise. It's fairly early on, there are definitely people studying it, but not a huge number. So yeah, there's a lot of future work that could go on there. Mm.
I don't know if you know this, but I've noticed that a lot of kiwifruit orchards are becoming enclosed with netting. Is it netting or some sort of like shade housing in a lot of places? I don't know if it's for bird or weather management or whatever,
Mm.
but I guess that might have an impact on the or even the access, maybe, I don't know, of spiders and other insect biodiversity.
Yeah, that's right. It's an interesting one because like some of that netting is very fine. So it could prohibit spiders. I mean, they can kind of crawl around it a lot of the time, but
Mm.
would they? Yeah, and then I think also if you go beyond spiders, it's going to have an effect on the invertebrate biodiversity in the orchard. So it's an interesting one. Yeah.
And it's actually quite, if you're looking at aerial views, it's actually becoming more and more. It's a big investment that a lot of
Mm,
growers seem to be going into. So I guess watch
yeah.
the space, but still, yeah. I guess we live in a changing world, as they say.
Yeah, that's right.
Um,
And I mean, there could be some benefits. It could provide some shelter as well. So,
mm.
yeah, be interesting to look at in more detail.
Do you think spiders for pest control works the same on a small scale as well as a big scale orchards? Wondering, like, as a gardener, should we be creating habitat to attract spiders to manage pests?
Yeah, definitely. I think it can definitely work on that small scale. And so having those structures in your backyard that spiders um, can use for their webs and not tearing down the webs all the time, but just appreciating those spiders and leaving them there in your garden, not getting rid of them. They will help feed on those insects that eat your vegetable gardens and stuff. So yeah, definitely. It can definitely work in your garden.
Oh, well, kia ora. I mean, oh, here we go. Gut content, kia ora, Vaughan. Gut content analysis sounds fascinating. Is this knowledge you already have, or can you apply it now, or do you need overseas trip for training? Is
Thanks
that
for
a
that leading
patsy
question,
question
Fawn.
for you to zoom off somewhere, or is it a legitimate
Yeah.
one?
Yeah, no, I don't know how to do this already, but I have some context. So there are people, there's like a handful of experts worldwide who do this specifically with spiders. And so yes, I would love to go offshore and learn this technique and be able to bring it back here to apply that and specifically for spiders, but it can be used for other invertebrates as well. But there are some technicalities with spiders because they are predators, as I understand it. So yes, an overseas trip for training would be great, Vaughan.
There's such things as postdocs and things too, isn't
Yeah, yeah. Well, I have
there?
applied for some funding. Yeah.
And we welcome Patsy questions before, in case I didn't think, but I think you know me well enough to know I love all sorts of questions, so keep them coming. In a garden, would encouraging spiders predominantly bring exotics? Do exotics take habitats from the native spiders or eat them?
Yeah, that's a good question, actually. I mean, it depends what type of plants you have in there, I guess, and also how close you are to like native areas. In the modified systems, you really get more of the introduced species of spiders. There are a few natives that do well in modified systems, but um, definitely the introduced species do better in modified systems. But if you have a whole lot of native plantings in your garden, and say you've got some native areas nearby, then you're likely to get native spiders in there. And all of the spiders do eat insect prey, so it depends sort of what you're looking for exactly. And yes, some exotic spiders do take habitats from the native spiders. You can think of the false katipo spider, for example, that is very distributed wide throughout New Zealand, and you'll see it if you have, say, a piece of wood in your garden and you lift that up, you're likely to have false katipo under there. Yeah, good question.
Yes, it's like, I guess, like the people that put bird food out and end up, they don't get little silver eyes or whatever, they get
Yeah.
minors, but still. Nelson, kia ora. Great work, Nicola. Is anyone looking directly at a design of refugia for web builders? I found it
Yeah,
stronger
interesting.
beneficial as I don't think they will do spider rearing, but
Yeah. Yeah, spider rearing gets a bit complicated because you've got to rear the insects to feed the spiders and everything. But I am not aware of anyone designing refugia for web builders. I know there is some literature offshore that has done some work on refugia. So yeah, that could definitely be a future project to look at that because yeah, I think there's good potential there.
kia ora, Teresa. Love your presentation, Nicola. Well done. Also very appreciative of your work in the kura and kaha. I would also love a copy of the insect flip chart, so that's really possible
Awesome.
and
Yes.
popular. And Vaughn's off.
See you, Vaughan. Thank you.
No, kia ora everyone. Thank you for coming and listening to that. Um, if you want to listen to it again or someone's missed it, of course, all of these will be online on YouTube for future reference, as are all the Rangahau Rising presentations. So thank you everyone for coming, and thank you, Nicola. That's been another fantastic presentation by yourself. I'll just put a pitch in for next month. Don't forget to hopefully the link will go in so you can register for next month's Rangahau Rising as well. And there's actually, there you go. Look, it's magic. So yes, if you want to go offshore, so we've gone into the realms of Shelob. Next month, it's the realms of Pacific climate resilience, agroforestry science, and public interest, presented by Dr. Suliasi Banabola, who's part of our BA Bioprotection Aotearoa's Pacific research. So that will be awesome. So please also register. But I'll close our hui. Any last requests, as they say, Nicola?
No, I think that's all good. Thanks.
Well, all the best for your postdoc and your trip to learn how to. Where is it? Is it somewhere exciting? That where they?
Europe would be the
Oh,
place.
okay, yeah, there you go. So no, thank you everyone. Thanks for the questions. It's great to see you all here and see you next month. And go safe, go well, as they say. Um, karakia whakamutunga e te papatonuku kua mutu tātou e te mahitahi o te rā kia tātou rangamārie ke mātou. Tihei Māori ora. Kia ora everyone.